This is TWIS, This Week in Science, episode number 996, recorded on Wednesday, December 4th, 2024. Why do we do the things that we do? It's not voodoo. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with trouble, insomnia, not like Justin, and tattoos. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. When humans do things, they don't always know why. They wake up knowing it's because of an alarm. They eat because of the time or due to hunger. They bathe, brush teeth, and dress themselves appropriately for the day or night with an eye on the weather. They work because if they didn't, there would be consequences. And they sleep because they know they need to be well rested before the alarm goes off again. Humans are, of course, slightly more complicated than this, but essential human attributes these days consist of lists of necessary routines. While they may be aware of needing to do them, they might not always know why. Some of the humans regularly tune into this show, where they jump into a conversation about recent research and fill their heads with sciencey news. But do they know why they do this? Or do they only know that if they didn't, they wouldn't know what they were about to miss on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin. And everyone else out there, welcome to another episode of This Week in Science. We are back to talk about all of the science that we want to talk about this week, that we think you might like to hear about and maybe have questions about, like we do as well. I hope everyone had a wonderful week last week.
Yeah.
I hope everyone is ready for another wonderful show full of cool science news. I have stories about glyphosate trouble, making muscles, microbe switch-ups, scalp tattoos, and feelings. What do you have, Justin?
Don't have any scalp tattoos.
No.
No. Is that where did I hear that right, or did I miss? Is that a story about scalp tattoos?
It might be. Mm-hmm.
My goodness. Okay. What do I have? I have new neural activity that's really actually pretty amazing story. Huge, groundbreaking, crazy, changing how we think or thought things work. I've got tired of being tired and lonely. Another Neanderthals did it first story. And what was there's a surprise story at the end.
It's going to be a surprise to you too, apparently.
Yeah.
It's going to be so much fun.
Yeah. What the ancient Native Americans were eating. So spoiler, it's mammoth.
All right. We already talked about it. No, we'll talk about all this on the show. I hope you're all looking forward to what is coming up. And before we get started, let me remind you that we do broadcast live every Wednesday, 8 p.m.-ish Pacific time. And that's on YouTube, Facebook, and Twitch. You can find us online looking for This Week in Science. Our social handles are at TWIS Science. You can also find us, if this stuff is just too much words going past you, go to twis.org. We've got show notes, we've got links, we've got other stuff. So I hope that we find you over there.
But aren't they already tuned in?
These people are, but maybe there are other people who just found us and are like, what's going on? And they haven't subscribed.
How
So I'm
wouldn't
like,
they know how they found us? Anyway, that's fine. Until we start broadcasting through
please subscribe, share, and help us
through
have more
cranial
listeners and
resonators.
followers. Tune in. Don't drop out.
If we can get one of those cranial resonators to just start broadcasting the show into people's heads, then I guess.
Mhm.
Then be like, where's
Yeah,
this?
I've got my own special satellite. You know, I've been working with the tech bros to get a satellite up there that aims TWIS.
It's how
right into people's
I
heads.
found the show in the first place. Yeah.
Oh my goodness. All right, let's get on with the science, everyone. You ready? Okay. So, in case you were wondering, glyphosate is bad, and it's still bad.
Wait. What is it?
Glyphosate. So glyphosate, we've talked about it on the show before, and I think the last time I talked about this compound, I got confused about what it was, and I was like, no, whatever. And then you reminded me that it was bad, and I was like, oh yeah, okay, that's really bad. Glyphosate is the main ingredient in Roundup, first created and brought into the public sphere by Monsanto, then later I think bought by Bayer. But anyway, Roundup, which supposedly now is no longer using glyphosate and is instead using some other cocktail of chemicals. And I'm not sure about what all that is, but I don't know. Get yourself some vinegar, dish soap, and salt, and maybe you can get rid of your weeds. This is an herbicide, right? So glyphosate is known to act on broadleaf plants and grasses. It's used very, very much in agriculture, and historical research has suggested that, oh, it's fine. It doesn't stick around. It breaks down and then it goes away. And people, if you're eating your food and you're doing whatever you're doing, you are not coming into contact with more than what is acceptable according to the EPA as an acute toxic dose. So, over the years, it has been looked at in terms of people using it as an herbicide. You're working in the fields. How much are you inhaling? How much are you, I don't know, getting on your hands or your skin when you're working in the dirt? How much is a person who buys a product off of a shelf at a gardening store and getting rid of their weeds actually potentially coming in contact with when they use it? And beyond that, how much is in the plant and would people potentially be eating or getting into their whole body systems, and what happens to it then? According to the EPA here in the United States, it's not toxic. It doesn't cause cancer. It's amazing. It still works, and they haven't banned it, but several other countries have banned it, and it has been indicated as a cancer-causing compound. And more recently, the evidence suggests that even though it may not build up long-term, and it actually probably does, we come in contact with it so much every single day in our water, our food, and the air that it is potentially that cucumber you ate is not the toxic acute dose, but you had a whole salad, you went to the river, you also breathed air. And they're finding glyphosate in all sorts of tissues. And this particular study that I'm talking about is brought to our attention through the Journal of Neuroinflammation. Researchers at Arizona State University and colleagues at the Translational Genomics Research Institute, Park City of Hope, they exposed mice to glyphosate to see how it impacted brain development. And the majority of research has shown that although you may get a significant amount of glyphosate and its metabolic breakdown products, there's a metabolic breakdown product that has the acronym AMPA, but it is nothing to do with like your memory receptor AMPA. This is a different molecule, and this particular AMPA, it does kind of stick around in the soil, and bacteria break glyphosate down in the soil. And there's a question about whether or not they break glyphosate down in your body. So and also in the plant itself. So you're eating a combination of glyphosate and this AMPA molecule, and these molecules inhibit and stop amino acid production. Glyphosate itself is supposed to get in the way of a particular pathway within plants. It's not supposed to affect people, so the people stuff is not supposed to be a problem. And the research has always been like, well, the glyphosate you eat, basically we see the stuff you eat, you end up excreting it. Your body supposedly gets rid of all of it. But there are minute amounts that apparently remain. And so work has continued to support this buildup and even small levels of exposure to glyphosate and its metabolites. And what they have shown is that with this experiment with mice, they gave normal mice and mice that were genetically modified for a predisposition for Alzheimer's disease. So no matter what, the Alzheimer's disease mice were going to get Alzheimer's disease. But it's usually it happens on a certain timeline and they know how it's going to progress. In this situation, they gave the mice a periodic glyphosate exposure, so that they gave them the glyphosate and took it away, gave them glyphosate and took it away. And then they had the mice 6 months glyphosate free, no glyphosate or the AMPA in any other food, no glyphosate in their environment. Supposedly, there should be no other way that glyphosate would be impacting them whatsoever. They found AMPA in the brains of both the Alzheimer's disease and normal mice after a 6-month recovery period. So mice supposedly, and mice it's a model for humans, but supposedly the glyphosate and the AMPA are supposed to be gotten rid of. They're not supposed to stick around in the body. But what we're seeing is that the dosage of the glyphosate and its metabolite end up in the brain tissue. They do travel. Glyphosate itself travels through the blood-brain barrier. It leads to increases in pro-inflammatory cytokine tumor necrosis factor alpha. And in doing this, it leads to a buildup of the proteins related to Alzheimer's disease and the symptoms of Alzheimer's disease in the mice. And so the Alzheimer's disease mice got their Alzheimer's symptoms earlier than they should have, but so did the normal mice. So there was Alzheimer's disease-like pathology in normal mice after 6 months, even though they should not have had that. This doesn't say this is what's going to happen in people, but we have seen that pregnant women have evidence of glyphosate in their brains. There's evidence of, this is similar to like the PFAS kind of stuff. This isn't PFAS, but there is evidence of glyphosate building up in the bodies and the organs of people. And no, you're not, unless you're taking a shower in Roundup, glyphosate is supposedly one of the safest herbicides when it comes to acute toxicity. But we're really beginning to start to see that the chronic toxicity is what is the problem, because glyphosate itself doesn't necessarily stay and build up in the soil and in the water, but microbes break it down. It turns into this AMPA metabolite, and AMPA is a little bit more likely to stick around. And so you get AMPA in the soil. And if you've got the same dirt year after year after year that you're putting crops in, you probably get more and more and more of these products into the soil that is where the plants grow, and our water and our air. And this is the happy story. Yay!
Well, and you're talking about it in terms of crops, but of course, this is one of the most commonly used like backyard chemicals.
Herbicides,
This
yeah.
is-
Which is why I mentioned that the product Roundup, supposedly, I do not know this for sure, but I found stories that suggest that it is now a different ingredient list, that it is not using glyphosate any longer, and that they are relying on different herbicidal compounds in a cocktail of them in order to do a similar job now.
But are we talking like, I mean, Roundup has been around for a long time, certainly. So are we talking it's changed in the last 5 or 10 years? Or
No, like in the last 2 years, maybe.
Okay, gotcha.
Because as of like 2021, the EPA in the United States still had not acknowledged, like the reporting and the research that the EPA did, they concluded that glyphosate was fine within certain limits, and that the way that people, if it's used properly, and so if it's used properly, it should be fine. But it did not. This is a really interesting, it's one of those places where it's like, okay, are we debating who's doing the study? Are we debating threshold levels? How is this all working together? But again, this is mice. It isn't humans. But it's a model system that showed elevated tau protein pathology resulting from glyphosate even 6 months after the dosing of these animals. And the animals were given a high dose that was similar to previous studies that have been used, because we know that when rodents and other animals are used very often, there's like the dosage is the poison, right? So there are high doses, which is like, maybe that's when this stuff is used badly and somebody drinks a bottle of Roundup, like whatever. Don't do that, by the way. But then they also had a low dose that was in line with the suggested concentrations of use by the EPA, and those animals at the low dose also had the same effects.
Okay, yeah, and my point though is also that the recommended dosage, when you get it into the hands of somebody who is just going into the backyard or spraying around a commercial property to get rid of weeds, this sort of thing, they're walking into it. This is a safe chemical. That's what they know. They may not have gone through and read every dose-dependent study regarding the usage of this. What they will go in thinking is this is safe. And if it's safe to spray it once, it's more effective if I spray it 4 times, right? Like that disconnect when we identify something as safe, as opposed to under certain conditions, this can be used with lower threshold or lower risk, lower hazard. Like it's a huge disconnect, and so I think that it's been a long time that we know, maybe you shouldn't be spraying Roundup around the family pets.
And I will actually admit, like for years, I was convinced by the science that I had seen that research had appropriately shown that the glyphosate compound was only acting on a metabolic pathway within plants, and that it was not going to be recognized or able to impact mammalian cells. Like we could probably go back over the last 20 years of the show, and I'm sure that I said that at one point in time. But the evidence is racking up that this stuff, it's not just plant sensitive or plant specific. This is having impacts that are more broadly impactful.
Yeah, and it's curious. I know there's like big class action lawsuits going on for the cancer causing potential right now. And I don't know. Yeah, I mean, there's going to be the lawyers will sue about anything, sure. But I don't know if it's because they're trying to connect it to the cancer, or if it's because that they have found that, hey, your company research, because that's like sometimes that thing that happens. It's like Oil companies are like, oh, we didn't know, but global warming was a thing
Oh,
or
well,
whatever.
yeah,
And then it
the
turns out they've
covering up
been
the data.
understanding it for 70 years and talking about it behind the scenes in their own research, right? Like, when a company has put
We
in the
don't know
harm,
this, and I'm not going to. We said some company names in this already,
we're not going to get
and so
sued.
I don't want to like make any statements that could be interpreted like we're saying that companies are covered. Like corporate stuff happens. We don't know. All we know is what's out in the public right now. And what I'm seeing is that the evidence is really starting to indicate that the EPA should be making some different decisions.
Yeah, but I know some of the studies that you were talking about, because they would do it with a 1,000-time concentration and spray it on like mosquitoes or something, and then you're like, that's not how, like, you could drown the mosquitoes with that like it would be totally ridiculous. Some of the studies that were attempting to show harm, and it's difficult in something like this, right?
Yeah,
the connections aren't, especially if it's not acute, it's the connections aren't for A to B. There's like this intermediary molecule now that we find is involved in the, like, it.
And one of the questions that they bring up is whether or not now, knowing that AMPA is this metabolite is causing a lot of this damage, as well as the glyphosate causing inflammation, as well as glyphosate. What do we know about how, and bacteria in the soil break glyphosate down? AMPA is a product of that. What are the bacteria in our guts doing? So we see glyphosate be excreted in various ways, removed from the body, but what metabolites are left behind? Nothing's ever 100% in biology. It's so messy.
And that whole like, oh, you can eat this molecule and 100% of it will leave your body. Also, just like nobody thinks that's true. It can't. It's like that never happens.
I think it's only true in that that one molecule that they used as like a fake oil for potato chips or whatever, Olestra. That one probably it's like Teflon for your guts. Anyway.
PFAS coated.
Yeah, but I mean, our bodies want to fight things off too. So I mean, what's happening with our immune system, Justin?
So this is probably the most insane new study this month. Gosh, groundbreaking even. Study published in Nature. This is researchers at the University of Copenhagen Imperial College London have identified a particular type of immune cell, the macrophages, that play a previously unknown role in how you move.
Wait, what? Macrophages.
Yeah.
Wait, no, macrophages are the ones that are like supposed to eat bad things, right? And get rid of macrophages are.
Yeah, it's the immune cells. It's they found them within muscle spindles, and they found them actively participating in motor control through fast neurotransmitter related mechanisms, typically have previously only been associated with neurons themselves. They seem to be working like a mediator of this neural activity within muscles. So, yeah, they're in a way fine-tuning muscle contractions in this interesting feedback loop with the nervous system. So there's a kind of a fun, the explanation in this press release, at least, is that if you close your eyes and take your finger and touch your nose, right?
Yeah.
Part of this is it's actually kind of a complicated thing. It's your brain is sensing where your fingers are as you're doing this activity, and they're slightly making these micro adjustments to get everything into the right place to pull off this activity. What they think is happening is that the immune system is actually helping that feedback loop of knowing where you are in that motion and communicating that to the brain.
How? What?
Yeah.
What? Okay, so we over the last several years have talked about astrocytes and glial cells, doing more of a short range communication between nerve cells. So we've got you've got your nervous system. You got the nerves that have these big long projections and the axons that go way, 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 way out down your legs or whatever, to go do a thing. And they're connecting to different stuff along the way. In the brain, you've got astrocytes, which are not necessarily immune cells, but they're like glial cells, the support cells. In the brain, they're doing helpful like waste removal and also helping with some of that communication. But this is macrophages in the rest of the body, in the muscle spindle. What are they doing there? What's going on? I'm confused.
So it has something to do with glutamate signaling. So this is a fast-acting communication system that is associated with nerve cells. And they think that this is, and it's also, central brain functions, memory, learning, movement.
Glutamate is the excitatory neurotransmitter.
So the macrophages use glutamate to modulate sensation and activate the muscles. So they're actually using it. So, and then the muscles produce glutamine, which reactivates the macrophages, having given up the glutamate, creating a continuous feedback loop. So the mechanism shows that macrophages' role as integrators of immune, sensory, and metabolic function in movement control. So,
yeah,
if this is taking place, which if this is a crucial factor in movement, there are conditions like stroke, Parkinson's disease, and even diabetes where inflammation, energy deficits, disrupted neural circuitry exacerbate the disease progression or indicative of it. Part of the characterization, targeting a dysfunction that we didn't know was there, something to do with macrophages, could be like a whole new target arena. The fact that we didn't even know that this was taking place.
Yeah. Okay. So I'm going back to my old school physiology stuff. And muscle spindles they're a part of the whole muscle bundle, and they respond like sensory fibers, and they respond to stretch. So when the muscle gets stretched, which would be indicating that there's a force on the muscle, the muscle spindle goes do 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 do. I gotta send a big thing, and there's a message that's sent out related to that. So what I'm guessing is metabolically, you have macrophages, which are immune. They're also in a space where muscles, when they're used, are damaged, and there's metabolic results from there's waste that has to be removed and taken care of. So maybe the macrophages are like sitting there hanging out with the muscle spindles, and when there's a lot of extra waste material coming off, the macrophages are like, extra, do extra work. You need to work harder. So maybe it adds to the signal. Maybe it's like a local amplifier.
I don't know.
I don't know either, but I think this is very exciting.
But it's like a completely unknown role of the immune system in the middle of what is a very common constant activity of muscle control.
But now it opens up a whole different, like a whole library of questions. You know, what are they doing there? Exactly. What is their role? Why are they communicating? How does it add to the communication process? What's happening? Justin, I want you to answer all these questions.
Oh, what is it's
where else do we find these, and where else do they work?
It's the Watson and Crick's first paper, which is actually an article. It's like a short, it's like a one pager for discovering DNA. Well, we understand the implications of this, and we're going to follow up in future papers.
Oh my gosh, that's like there was an announcement about a big supercomputer work doing the first simulation of an area of the visible universe. Mm-hmm. Like it's the first time because they're using exascale computing. So the press release was like, we're just going to use the fact that we did this really cool thing as a lead in to all of our exascale computing stuff. And at the end of it, the whole article is like just glossing over this amazing feat of actually being able to include all of the parameters that are known that are part of the universe and that impact the universe for the first time. You know, previous simulations on supercomputers, we've had to just kind of leave things out, and it's just general. Now it's like a finer resolution, whatever. No. Look, exascale computing. We'll be back to you later. And that's why I wasn't going to bring that story to the show tonight.
But isn't it always exascale until like...
No. Remember, we had a whole interview on this like a couple of years ago. It's like the new exascale computers. It's like it's not supercomputers. It's exascale. It's bigger.
Yeah. But then there'll be another... I feel like there'll be another scale after that.
Yeah.
As the computers get better, and as we get more rich in our own data understanding of the universe.
Of course there will. Yeah. All right. So I'm going to stick with muscles for a moment, and I want to talk about a new study that researchers were trying to figure out. You know, they're looking at all the genetic factors that are involved in and also the epigenetic factors that are involved in creating muscle. And so the study, they had two phases where in the first phase, they had exercise happen. They made cells stressed out. They looked at all of the genes that were expressed, and they went from like the genome to the proteome. What are the things that are being expressed? Also, what's being wrapped up in sugars and held in so that they're not being transcribed and translated? And then what makes it to being a protein? So they did a whole range of analyses, starting out with hundreds of possible factors to look at. Hundreds. They're all working together, and they all have massive numbers of downstream effects because this little protein gets formed and that protein goes and triggers a thing. And so it's like this cascading scale of things, but they were able to analyze the whole system, looking at it methodically to determine there's one particular gene that to date, you know, we've talked about it all being related to cancer and tumor formation in muscles. It's a gene called MYC in skeletal muscle. And so this, the second half of the research used mouse models to genetically turn MYC on and off using a signaling dosage. So they pulsed production of MYC in mouse skeletal muscles, and they saw that it stimulated muscle growth without any exercise happening. So Will we one day be able to help people who are unable to exercise, who cannot move their muscles, whose muscles are atrophying because they are bedridden, because there is no ability for them to do movement, to be able to maintain their muscles, which also maintain their skeletal strength, which also maintains so much of the health of your body? So this study suggests that MYC is a target that we could start looking at, and also its downstream targets could be addressed in the search for a therapy or a way to build muscle without exercising. And yeah, of course, somebody's going to misuse it at some point if it starts being publicly available. But I think this is really cool. So the researchers, a researcher, Ronald Jones, PhD candidate at University of Arkansas, worked with the Karolinska Institute in Sweden and the Linköping University in Sweden, and also University of Kentucky. But Mr. Jones says that we show that the peak of responsiveness and where most things were happening was actually 8 hours after exercise. So they found 3 hours after exercising that MYC ranked as the third most important molecule. And then at 8 hours and 24 hours, it was the most influential. So they did this long-term time expression to see how exercise influenced not just that moment, but down the line. What happened with the muscles, the genes that were expressed to create proteins, and those proteins influencing other proteins, and what that did. And so they were able to map out the body's response to acute exercise, and then target a particular molecule or gene that can be used in the future.
Yeah, well, I think this is actually going to become very important because
when we're in space.
No, oh yeah, that's actually that's fantastic. That's like yeah, that could be huge. But I was actually thinking because we have now weight loss drugs that people are taking that don't require exercise. Yeah. So,
and they're actually very interesting and have a lot of interesting, potentially healthful effects, right? There's pros and cons for sure, but
I don't know. They've been out for as long as Roundup has been safe. So I don't know. I have no idea.
That's funny.
But it is all right. Like what was I looking at? It's I won't use the name because we're afraid of getting sued by everybody now. But a popular program that helps you watch your weight has also begun selling this as part of their programs, which I was like looking at that. I was like, isn't this sort of like if Blockbuster was selling Netflix subscriptions like on the side? Like, don't you see that's anyway? But yeah, like if you're taking a pill instead of exercising to lose weight, and so, but you still like, how do you then like have that part of your activity that's building muscle, that's reinforcing that whole, the skeletal organ.
Right. But this is what they saw is that those mice that were not given the running wheel exercises, but were artificially stimulated in a way that made this MYC gene make a protein, cause all its stuff downline. They built muscle.
Yeah,
even when they should not have. So, but the question is, if that process is taking place, but the stimulation is only impacting one particular gene as opposed to all the other complex of genes that are usually activated when you exercise normally, are you still going to get the same benefits that you get muscle buildup, but do you get all of the same systemic health benefits that actual exercise creates?
And then also
mental health, everything, right?
Like, I've been taking this, but it turns out it only does leg day. Like Suddenly I got these strong legs, but I still have the
Right. So
biceps. Like,
right.
how
So
did
they did look at different muscles to see how they were impacted, and they did see a small difference between different muscles in the activation of MYC. They don't know why some were activated more than others, but generally, generally all of the muscles they looked at reacted and built muscle. But they did see that some muscles built muscle more than others. So and they have not yet tested this in humans. This is just mice. So you're getting weirdly like gluteus maximus or soleus, whatever mice, mice with really big calves.
It's all butt muscle. Like, that's the only thing that because that seems like a little bit strange. Like, like at least with a lot of the other performance enhancing drugs, you still like would get, it would all be concentrated on, your whatever reps of exercise I assume you were doing. It wouldn't just be random muscle placement throughout. Like, oh, yeah, it turns out I just all worked on jaw muscle on me. Yeah, I got these big jaw muscles now, but didn't do anything else.
Yeah, so one thing I thought was very interesting is that the compound that they used to create the MYC stimulation is actually an antibiotic. So it's wait, was it dextromethorphan or no? I'm scrolling down my paper here and I'm going to get it. Okay. No, that's right. Doxycycline inducible muscle specific activation. So it's a combination that has been used in previous experiments, apparently, to cause overexpression of certain genes. And it's been used in some of the, Shimonaka, like gene stuff that creates influencing genes in different ways. But it's called HSA-MYC, human skeletal actin reverse tetracycline transactivator tetracycline response element TET-on MYC. Anyway, the animals drank doxycycline in their water, and then they had 12 hours of no doxycycline in their water, and then they drank it again for 12 hours, and then no doxycycline. So it was this pulsatile use of doxycycline. Now, this is what I find very interesting because we use doxycycline as an antibiotic, a general antibiotic, and it's fascinating that it is also being used for gene activation in experiments. So I have questions about this that I cannot answer at this point in time.
If there's like a certain like mediocre dose that you could just take for people and they wouldn't have to exercise, I think it'd be great. Like exercising.
No, we all need to exercise.
Like the most ridiculous thing. If you go like seeing people run in place.
Exercise can be walking.
Sweating.
Exercise can be walking up and down the stairs in your apartment building. Exercise, we say exercise, but it's really, we say exercise because people need to move. And you can exercise extra. I mean, I want to redefine it as extra size.
Extra. Okay.
All right. We'll move on from my doxycycline muscle mice. You want to tell me about college students?
Feeling tired.
Oh, I'm tired too. Oh,
tired and lonely, maybe. You aren't alone. I mean, obviously you are alone, but you aren't the only one who's alone. Have you considered that maybe you spend too much time in front of a screen? Yeah? Well,
never.
You're wrong. Yeah. That's just a symptom, maybe. Not necessarily the cause. It may be true that you have too much screen time, but the real reason you can't sleep and are so tired is because you need a more active social life. Huh? Well, it turns out that being lonely is a bigger hurdle to a good night's sleep than too much screen time. New study by Oregon State University suggests. Insomnia and loneliness, these are serious public health concerns that are at what is called epidemic levels. Especially for young people. Like this is like college student age, higher education, young adults apparently aren't getting enough sleep. A study of more than 1,000 undergraduate students found that when an individual's total daily screen time reached the 8 to 10 plus hour range, there was an increased likelihood of insomnia. So maybe it is that screen time. They also found 35% of subjects had high levels of loneliness, and that lonely students were more likely to have trouble sleeping than less lonely students, regardless of the amount of screen time. That 35% reported clinically significant symptoms of insomnia at almost twice the rate of the other 65%. That's pretty huge. That 35% were twice.
It seems like it's a big difference.
Yeah. Screen time was definitely associated with insomnia symptoms, but loneliness was the much better predictor of it. And this is kind of like becomes like one of those what came first. Like if you're spending 8 to 10 hours of screen time, maybe that's eliminating opportunities that you might be having, social interaction during those hours instead. But
was it that you didn't have social interactions and so you went to the screen time, or is it also that you had? Is it also we're having issues with this before the pandemic? But is it also that, oh hey, you spent 3 years having to do things online, and now you just do things online?
Or it's like these are college students, so it's like they spend all of that time working on papers.
Yeah. I don't know. I find this very interesting. I would love to know the difference in loneliness and insomnia research before digital media became a thing. Like how much more were researchers looking into these questions seriously?
Yeah. So, but college student or not, I think the moral of the story is get out there and mingle, meet some folks, talk about whatever. Go have a social, but not right now, because right now it's 3 a.m. You haven't
No, it's 9:15.
Not the time to go out and find people and start talking to them. This is not the right. So you got to find the right scenario condition.
What I'm hearing here is that the treatment for college insomnia is to go out among people because people are tiring. No. Is that true?
Well, then I'm thinking about like, what if if you didn't have any screen time and were completely social and still didn't sleep, then then there'd be no hope.
But that's the thing, right? But this is the 35%, double the rate of the 65. This is that important number. So it's screen time does affect insomnia. We know that blue light, we know stimulation, we know all sorts of things do impact sleep quality and insomnia. Maybe you're using screen time as a distraction, a deflection, a way to I'm having bad grades and I'm going to just dive into the internet instead of actually dealing with stuff. It's not going to help. And then you try to sleep and your brain's like, what did I do? Oh my God, I'm a bad person. I'm not speaking from experience at all.
That was different.
Sleep, you're like super stimulated. Like, why can I sleep? I should be sleeping. Why am I not sleeping? And then you think about it too much, and then you can't sleep because you're thinking about why you can't sleep. And anyway, feedback cycles are amazing. But if you were to, maybe there are mental health issues that go into this in the first place. So there, like you said, chicken egg. What is the thing that leads to the thing that lends to this? But in addition to screen time, contributing to insomnia, loneliness is a stronger factor. And loneliness probably also contributes to screen time.
Yeah.
Maybe.
I would. Well, in this day and age, absolutely. Yeah. I mean, the other confusing part is you can have screen time and be hanging out with people. So
yeah, my child does that a lot.
Yeah.
He seems very happy. He's like, I'm having a great social life. And I'm like, do you know these people? He's like, I don't know. I'm like, okay. I'm worried about the modern world. What do we do? Well, in the case of researchers, they give mice human bacteria. So why not? Researchers wanted to know how the human brain got so big, and we have talked about the microbiome on the show many times. There's been a question as to whether or not, certain nutritional aspect, behavioral aspects, what happened historically, evolutionarily to lead to the change in our brain size and the way that it develops in comparison to other mammals. So this study, which is out of Northwestern University and published in Microbiology Society, Microbial Genomics Journal, says the primate gut microbiota contributes to interspecific differences in host metabolism. What does that mean? Well, when you give mice human bacteria or other big-brained primate bacteria, it changes the way that they develop and the way that they metabolize nutrients. Mm-hmm. So the take-home message of this study is that, unfortunately, it wasn't completely a Freaky Friday situation. They didn't give any humans or primates mouse microbiomes, but they took sterile mice and gave them human microbes and also like large primate microbe, and then other groups got small primate microbes, and they demonstrated that the gut microbiota of larger brained primates shifted the metabolism of the mice to use energy differently. And so smaller brained primates stimulate energy storage in adipose tissues. So it's like larger brain primates shift metabolism to use the energy, get it done, grow big now, do things, yay. And then the small brain primate microbes were like, let's put that away for later. We might need that for our energies. So even though they had human and a large brain primate and a short brain primate as the donors for this, there was no difference, or relatively no difference, between the human and the other large brain primate on how the mouse metabolism shifted, which is kind of interesting and very cool, because this is one of the first studies that has kind of started to parse out how the microbiome influences metabolism, how it influences the utilization of resources, and also development of brain and body and all that kind of stuff. And it's been a question for a while. So I like it. It's cool.
But does that mean that you'll have the reversed effects if you just persist on a diet of mice?
Don't eat the mice. Well, no. If you eat mice, yes, you're going to eat some of their microbes, but you're also a human. So you're going to be living in your human world with your human microbes. So you got your microbes there.
Yeah. Okay. Well, I don't know. Maybe. Yeah. I still my favorite one is the microfluidic pump. That is that by having fleshy feet, when we were walking and then running, every time it would pulse like the brain with a little extra blood, and that over time, as we walked more and ran more, the opening got bigger and the veins got bigger, so you could pump more blood to that brain.
That's not saying that's not part of it. This is just starting to get at it's all part of it, but this is the microbes, right? And so when you give mice human microbes, the way that their body works changes. Right?
Yeah. Yeah.
Kind of interesting. Anyway, I think it's interesting. Do you think it's interesting? I think it's cool.
It's all very interesting.
Freaky Friday Mouse.
I need to say that in a snarkier way, though. That's going to be a good. That's my new tagline.
Snark?
What is it?
What's your new tagline?
It's all very interesting, but it's a very snarky.
I'm scowling at you right now. Oh my gosh.
It's worth it.
If you think this is all very interesting, please head over to twist.org and click on the Patreon link and help support the show in an ongoing way. We love your support so that we can keep coming back week after week to talk about science and talk with you about science and listen to what you want to say. I do read all of your stuff. I try to follow along as you chat in the chat rooms. Additionally, if you're interested in holiday things, you now on Patreon can gift people Patreon subscriptions to TWIS. Additionally, there's a link on twist.org to go to our Zazzle store where you can find fun gifts, mouse pads. Like, I don't know, would you give someone a mouse pad for Christmas or Hanukkah or what? I don't know. Anyway, buy yourself a gift of TWIS. Honestly, I don't like asking for all of the help, but it's the thing that has to be done. And of course, this is the holiday season, so it's, commercially the time to ask.
Or you could just mail us money. Like, what is this intermediate thing? What are you talking about?
We have a PayPal link on the website as well. You can do that. You can mail me. No.
You find extra money lying around, but don't just mail us coins, because sometimes they can work their way out of the envelope as it gets moved back and forth.
I'm not interested.
So wrap those coins in some kind of a dollar bill.
Cashier's checks only.
Actually work better.
Oh my goodness. Honestly, I love you guys so much. Thank you so much for being here. Please share the show if you love it. That's the best gift you can give us. And I can't do this without you. We can't do it without you. I can't do it without Justin either. I couldn't start the show.
It would actually be a very different show. Like I'm already thinking of, if we were a sponsored show, there's already 2 stories that we did or things that we talked about today that we'd be like, nope, can't do that one. They're a part of the sponsor network, or they own our sponsor, or they're whatever.
That's a great point, Justin. Honestly, we are not commercially sponsored. We do rely only on listener support. So it's thanks to you that we're able to keep going and keep putting this on the air. Thank you for your support. We cannot do it without you. I mean, we could, but it'd be very, very difficult and boring. All right, let's come on back to some more twists right now. And I think Justin has some stories. Do you want to talk about Neanderthals or mammoths?
Actually,
or neither?
Actually, those stories could be a both, but it's not.
Oh.
So
okay.
I'll start with the mammoth story. We have our first direct evidence that ancient Native Americans feasted on mammoth. Well, we kind of knew that, but this one is showing that we relied primarily on it. Which, if you take down a mammoth, chances are you know what you're having, not just for dinner, but for breakfast tomorrow, for lunch the next day, and for what's leftovers. Leftover
bigger
mammoth.
Than a turkey, that's for sure.
Yeah, leftover mammoth for weeks, depending on how many of you there are in your group. Okay, so this study used stable isotope analysis to model the diet of a mother and infant discovered at a 13,000-year-old Clovis burial site in Montana. This is one of those things, too, which is very controlled by treaty in the United States, where you can't just find Native American remains and test them. You have to have the okay from the local tribes. Otherwise, you're not allowed. So this group in Montana has been allowing for research into the burial site they found that the prehistoric diet inferred by analyzing secondary evidence like hunting tools and marks on fossils. I mean, this is how we've previously done it. We've looked for all the implements and, found the fossilized carcass remains of a mammoth, and there's human spear points sitting in and amongst like the rib cage or whatever, right? So this is how we've determined that these hunts went on previously. But these findings now are somewhat unexpected in how much they relied primarily on mammoth. So
So it was a lot.
40%
What?
of this mother's diet, 40% of her diet was mammoth. There
Jerky?
was the other 60% was other big game. So it would be like bison or elk or a mixture of other sort of megafauna from the day. And we kind of knew like that these were big game hunters, right? Because of the tools, like the weapon, the spear points that are detachable, all of and the size of them, like none of what the Clovis culture was making made sense or would have been efficient for going after small game. Like we knew their tools were big game. So we've kind of assumed that they were, doing, but for 40% of the diet to be mammoth, so
I wonder if it was a fact. I mean, they're so big, so each mammoth taken down would be a significant amount of food. But then, if they had figured out how to preserve it, cook it, make it jerky, it could last a very long time.
Or if the climate's just cold enough,
As a frozen mammoth, right? You bury it. Yeah.
Bury it, boil. I mean, you're going to have to get creative anyway, because you're going to be eating mammoth for a long time.
Isn't there a thing where it's like a dry preserved steak where it's not like jerky, but it's been kept in salt or something for a very long time, and you can get these steaks that they're not jerky, but they're basically like a month old or 2 months old or whatever.
Yeah.
It's a whole process that I know nothing about because I don't eat that kind of meat. But
salted meats, that seems like a preservation technique.
Yeah.
But it depends. I couldn't find it, but there's a ratio that was done in actually some Neanderthal research where they were doing that ratio of the amount of time this food would last for versus the population size, like how many people it takes to eat a mammoth and how long that would take, depending on, right? So if you have, a large group, and it doesn't have to be like the hunting parties tribe, but then you're put the word out that we got a mammoth, invite all the neighbors over, because we say we only have 2 weeks before, where our salted meat strategy isn't that advanced or whatever. And so now we've only got a couple of weeks or days, whatever, I don't know, to polish off this mammoth.
But if that's the case, you feed everybody, you have a big party, and then the meat's gone. But then how does 40% of your diet end up being mammoth if it's these really short things? So obviously they're following mammoth herds. They were, the hunting was consistent to bring down mammoths.
Right. So if you have the preservation technique that's allowing for the 40%, so we don't know if the 40% is just because they were taking down mammoths all the time, or if that was good, you take down one mammoth and you're good for 2 months.
Yeah.
It's only 6 a year. Yeah. And it's only if 40%, and the rest of the time you're picking up a deer here and elk there, bison or what have you. Yeah. So this is this is also younger dry. So this is couple of things already start jumping out. First of all, we look at the Clovis culture was very, call it nomadic, but it looks like what they were doing is very likely following migration of mammoth, as mammoth changed seasonally from one region to the next to take advantage of different biomes as they appeared, seasonal ones. The humans might not have been as nomadic because typically elephants go to the same spots all the time. If mammoths are doing the same sort of activities, returning to the same grounds, going back and forth along the same routes, doing this sort of thing, those human migration, the mammoth migration routes would have very much become human migration routes. And so you would have sort of settlements like, oh, we're at this part of the Great Plains this month. We know a good camping spot that we always use over here. And so you get these sort of settlements that start to build up over time.
And they're more regular settlements. It's like some years maybe they're not great, but people return to them over and over.
So the younger Dryas is this crazy event where there's huge fires that spread across North America, likely from some kind of meteor strike in Greenland or something like this, right? This is the same time when all of the major megafauna sort of disappear. And so that's sort of considered how that happened. But now that we're seeing 40% of the diet being mammoth, I'm much more... I need to know what those numbers are. I need to know what that ratio is. Like how many
the other animals are other things, right? They're...
The other animals are just too fast.
Bison, Butherium, camelox, cervix.
North American camels.
Equini, so some horses, ovis, rangifer, small mammal. Yeah, so there's a good mix of a lot of these things. But this one individual mother that they were looking at was definitely mostly mammoth. And then Antilocapra, so like related to antelopes.
Yeah. So it also brings up some interesting stuff too, because
elk and bison.
What is it? What is that site in? Well, I don't have it in front of me, so we shouldn't talk about it. Somewhere in the coastal Southern California, they found what looks like cut marks on mammoth bones that were, 200,000 years old? It's like way out of the scale of when current modern humans are supposed to be around. But yeah, Neanderthals hunted mammoths. Mammoths were feeding every human.
And big cats, like saber-tooths and others. So I don't know. There's the whole idea that, humans killed out all the mammoths, but it was also climate change and everything. But it was probably humans were very good at hunting and they did a great job at feeding themselves. And there's probably a lot of like hunting knowledge that Western culture has just not paid attention to.
Yeah, now, did we discover earlier during this episode that eating a larger mammal, or no, having the microbiome of a larger mammal changed the way that you develop?
That's not eating their meat.
Maybe
No.
human. Yeah, but we're going to interact with the microbiome big time. You're opening up the...
But there's a difference between having no microbiome and then having an organism's or individual's microbiome put in you versus I just ate something. We eat lots of things. You're not developing like a cow or a piece of corn. I mean, come on.
Because you're not interacting with their biome, their microbiome. You would be if you were rendering a mammoth, following mammoths, stepping in mammoth dung all the time as you track them.
So you think
totally all up in it.
mammoths did not give people big brains. People already had big brains to take down the mammoths. I'm
starting to wonder. It's all connected, man.
It's all connected.
What was I've already forgot my tagline.
I'm
It's
not going to
all
remind
interesting.
you. Yeah. All right. So from mammoths to what?
And then this is
You have a whole other study.
yeah, I forgot. This is a fun story. This is actually the second or third version of this story that I've heard or seen over the last few years, but this one is slightly unique. This is a discovery deep within a cave in Spain that is challenging the history of human artistic expression.
I
Researchers
don't think
have,
I've ever heard you say this before. Actually, no, you've said this before. Why is this unique?
researchers have
Right.
determined that hand stencils in Malta Rovescio cave are more than 66,000 years old, suggesting that no current modern humans didn't make these. These would have been from Neanderthals. So this is very classic stenciled handprint, right? Where the hand is placed on the wall and something is used to spray or spit or brush the pigment around where the hand is placed. And then you take the hand away and you have the stencil.
It's how kids make like turkey decorations for the Thanksgivings. Yes.
The Malta Rovescio cave houses more than 60 of these hand stencils. But the precise age has been a mystery. And this is always a problem with not just rock art, ancient rock art, but a lot of art is that it's the things that are going to last through the ages that are going to be painted onto rock walls are going to be mineral based. So if it's mineral based, you can't use carbon dating because that's something you use on organic materials. So what they've done is they have used U-series dating, which is, we've talked about on the show before, on a painting, the red dot artwork that we see also in Spain. Thought to be early human, but then got U-series dated to earlier than 66,000 years ago. So U-series is uranium converting to thorium, and uranium decays into thorium, and types of thorium decay into other types of thorium over a very specific half-life tick-tock atomic type of time. You can set a clock to this from a couple of 100 years to 500,000 years. It's actually
which is great for
one
our
of the
evolution.
biggest time span things that we could do. Now, usually in most places, it's totally useless. You picture like the surface soil, you have just environmental uranium isotopes landing all the time. This can be emitted from, in modern day, it would be emitted from coal plants, but it's also volcanic activity, atmospheric dropout of these uranium isotopes end up in the soil. And it's just such a mixture, hodgepodge in there. You can't tell if this thorium came from that uranium, or that uranium produced that thorium over there. So you can't age dirt, right, on the surface of the. Now, what's interesting is the uranium isotopes are water soluble, and the thorium ones are not. So it rains, and that the uranium is able to sort of move around in the water as it moves. It can hitch a ride. And if there's a cave structure underneath and the water is working its way through, that moisture from above works its way into the subterranean caves, maybe comes dripping down a wall, and then the water evaporates and it leaves behind a collection of minerals that it has brought on this journey. One of those minerals that's brought on the journey is the uranium. And then over time, it forms these layers, this crustation, this calcium carbonate crustation forms on the wall of a cave. And because the uranium has been isolated from all of the other thorium, it starts that tick-tock conversion into the decaying into thorium at a very set pace, isolated within the layers of this carbonate. So, U-series dating, they'll drill out tiny bits of that crust that might be covering a certain area of a cave, and they can tell how old that crust is. And in this case, that crust is forming over some ancient cave art.
Huh.
Then they can tell, the pigments that are on that wall are older, of course, than whatever was dripping over in front of them over many, many thousands of years and forming this crustacean. So by just dating the crustacean,
I know you keep saying crustacean, and I'm like, what? There's no crustaceans in the cave.
There's no crustaceans involved.
Yeah, no
crustaceans were harmed in this research.
It's a crusty surface.
Yeah, by the calcium carbonate sediment cemented over the artwork. By dating that, you can tell that whatever's behind it must be older than the thing you've dated.
Mm-hmm.
Now, they had a whole range of dates from different areas of the cave over different pieces of artwork. Some only were, it had to be older than 6,000, 10,000. But there were 3 dates that were over hand stencils that are older than our oldest hand stencil outside of this by 15 to 20,000 years.
Because everything that we have
25,000
seen, everything we've seen as a hand stencil is an obvious hand stencil so far. These are like hidden, right? Are these not obvious? Is it that we can't really see them, or?
No. It's the oldest ones that we found before are from Indonesia. Like this is what's also fascinating about this is that this is like a common rock art motif around the world.
But what I'm wondering about these particular pieces of art that are part of the study and what they're showing and what they're establishing the age of with the dating, like they show that it's a red rock wall. It just looks like a cave, and there's very vague impressions that you can see maybe that are hands there. And then there's a false color image that shows super obvious hand stencils. But every time I've seen images shared of historic stencil art or art, it's been obvious. It is not false color. It's just a picture that you can see. It's like, oh, somebody took an outline of their hand or whatever. So I'm just really interested in what we're seeing in the images and what is actually on the walls and how that changes the way that we can look back into history by looking at like the different minerals, the dating of the minerals in this crustal way creates an image that would not otherwise be visible, right?
No.
Am I wrong?
Yeah.
Okay.
So sorry. So this is a non-destructive dating technique, is what they always refer to.
Oh, for sure. Yeah.
Because you aren't actually dating or contacting any of the pigment. It's just in front of, because it's over the pigment, you just can date that crust and know that the thing behind it's older. And the oldest U-series date that they got was 66 point something thousand years ago. Yeah. Meaning that the hand stencils that were behind this, they are older than when current modern humans are thought to have been anywhere in Iberia or Spain, right? They are at a time when we know Neanderthals were there. This is also at the time point where another cave that I think we talked about, this would have been like 3 or 4 or 5 years ago we were talking about on the show. They used the same methodology to discover that that sort of famous red dot artwork that I always think is like a play. like a strategy, like they've written up on the board. Shows a guy waving. It's got like a bison facing one way, a lot of red dots. So that one was also in this range of over 66,000 years. Now this could be 100,000-year-old stencil. We just know it's older than 66. It could be much, much older than that, even. Because the dynamics within the cave are going to change. Water flow and humidity in the cave will change, and where this moisture falls is going to, what rock walls it changes over, a course of 60-something thousand years is going to change. So even the artwork that's on the cave, it could be that current modern humans moved in, 10,000 years ago, saw hand stencils, and were like, oh, we know, let's try to make one of those. How'd they do that? Oh, or those are, also 40, 50,000 years old, but but they've only been getting covered or having the carbonate crust placed over them over the last few
tens of thousands of years. Yeah.
But the oldest previous to this of a hand stencil is from a cave in Indonesia that dates same type of dating this style, series, and that one's 39,000 years old. But again, because of the method that's being used, that is not dating how old the thing is. It is putting a minimum age. Just the minimum age. Could be much older. Cave in Indonesia, 39,000 years ago. Now we are in Spain, 66. So this is also, I think, the oldest cave art. Also, I think there's also the dots. It's the oldest hand stencil, at least, ever recorded. And
it's not just random stuff. It's like on purpose. People did this for a reason. We don't know what the reason was.
Some of it looks.
They went back in a hidden cavern in the back of the cave, and they're like, "I'm going to put my hand here."
Yeah, it looks like some of it, like that one picture. I sort of picture as the art you hang up in the bedroom. It's the art that's all the way back in the furthest recess of the cave that may not be as obvious to. But then there's the stuff in the front where you're like, it's like a little studio. But this is also. The red dot one that we were talking about a few years ago was fascinating because that had been thought to be current modern human, and it showed through U-series dating that it had to be over 60,000 years old, which puts it in the Neanderthal category. Here we have another example of that where we have shown that these hand stencils that we thought were current modern human had to be much older, had to be Neanderthal. Now the red dot in this.
Can I bring up one?
Pigment. Hang on, let me just finish.
Okay.
The red pigment used here and the red dot, finger dot stuff. Those aren't just in these couple of caves. They're in hundreds of caves throughout the region. They just don't all have the luck of having the carbonate crust in front of them to date them. But what it indicates is that cave art was sort of a ubiquitous, common thing that Neanderthals were engaged in. They were making art. And we have the 66,000-year-old art because it's minerals on a cave in a controlled environment. What we're missing is all the canvas work, right? Because that would have deteriorated. We're missing all of the other, artwork made on wood or carved wood, or like a lot of these things that would have deteriorated out of existence by now.
Kevin Unique in our Discord chat says, well, let's speculate about why those are left hands. And it's actually very interesting. The predominant handedness of the hands on the walls in the images that they share in this study are left hands. There are, though, a few right hands, but not that many. So if you look through, I mean, who knows? I don't know if they have counted this yet, but I think that's a fascinating thing. There's one, is it a right hand, a left hand in this one image that might be missing a finger? I don't know.
Yeah, the pinky's just.
It's fascinating. The pinky, like the little toe, it's just going away. But I think those are very interesting questions.
So that would indicate to me right-handedness.
Right. which
Yeah.
is still the predominant handedness of humans today. So Neanderthals maybe were right-handed as well for
They were right-handed. Although, it turns out they may have been
Interesting.
left-footed. So in this next story, I will talk about how they determined if left-footedness. No, that's not. That's not.
I didn't think. Wait, I was like, wait, that's not in our rundown. What are you doing?
I think my son might be left-footed. Actually, he kind of does things with both hands, but
I'm left-footed.
He mostly favors right. But whenever he goes to kick something,
I'm
it's the
left-footed.
left foot.
Mm-hmm.
That's wild. I didn't know you could be.
In gymnastics, I was a lefty. I always did left-handed cartwheels, left-handed walkovers. Everything I did was left-handed, but I'm right-handed when it comes to writing and all that kind of stuff. When I heard that was weird, I was like, oh, that's very interesting.
Oh, it is weird though. See, I was for a second. I thought, oh, my son isn't a freak, but now you're telling me.
No,
Indeed.
But there are hypotheses as to the left-footedness being a thing because of the foot that you land on, or what you do is the next foot is your right foot. So the stability that allows you to use that foot is your right foot. So there are ways we can rationalize everything.
There's
it's
no
also
foot stencils
interesting.
in any of this artwork. Why is there no foot stencils?
I think you need to go to the caves in Spain, Justin, and look for foot stencils. Where are the feet on the walls? Nobody had feet. That's all.
There is Tibetan Plateau, 200,000-year-old, I think. Yes, 200,000-year-old handprint and footprints that were made by children playing near a hot spring. Yeah. So there's some
old.
And that's, I don't know if you call that art.
I do.
People are debating that, but yeah, when kids are like, "Oh, look what I can do," that's probably the first artwork. 200,000-year-old handprints, footprints, slapped into the mud.
Well, talking about our old ancestors and, how similar we are to them and, what games we played and how we did things and how we felt, researchers from Aalto University have just published their work in iScience on texts from ancient Mesopotamia. This University of Helsinki researcher, an Assyriologist, led the project, and he says, even in ancient Mesopotamia, there was a rough understanding of anatomy. For example, the importance of the heart, liver, and lungs. And so what they did is they analyzed ancient texts from 934 to 612 BC in the form of cuneiform scripts on clay tablets, which were all sorts of writings describing everyday activities, business arrangements, all sorts of, various things. But these are these clay tablets from thousands of years ago.
People always focus on the tablets. What I always found interesting is the sturdiness of their bookcases.
What bookcases? Oh my God.
They had to hold some serious weight.
Well, with all that weight came a lot of the weight of the emotional baggage they probably carried. And so these researchers tried to figure out what words were expressed along with body locations and an expression of how they felt their emotions in ancient Mesopotamia through the writings that are left to us on these old clay tablets. So first, they're having to translate the cuneiform, and then they had to make a comparison of words to matching words for today, try and figure out body parts and emotional words that all correlated. So, there is a, disclaimer that, we weren't there. We didn't write this stuff. We don't really know this for sure, but this is their best guess for how they expressed their emotions back in the day. And whereas we talk about our anger or fear as something that our anger is we feel emotions in our chest. Very often, the Mesopotamians felt their anger in their feet.
They felt their anger in their feet?
Anger was in their feet, according to this study. Emotions
what was
like
the quality of footwear at the time? Because I can understand that.
modern humans also express it as like it's our upper body or our hands. That's where we're. They felt Mesopotamians, the words that they have translated, heated, enraged, angry, it was in their feet. Love was associated with not only the heart, but also the liver and the knees.
The liver?
And the knees.
I can get the knees. You get a little weak in the knees.
Weak in the knees. Okay, yeah. Okay.
It's like, oh, yeah, she's really got my liver excreting. Like, I don't know what I wouldn't even know I had a liver. if I hadn't learned it, I wouldn't even know it existed.
They did, apparently. They're like, my liver. Oh, my goodness. But yes, so they were able to approximate word vector values and similarities to various parts of the body. And they created these body heat maps for each of these emotions. And so they have a whole bunch of different emotions that were related to various ideas of emotion. And so happiness, it's the liver.
No,
everything's in
not the liver. I'm telling you, I haven't read this, and so I'm being very arrogant, but I'm telling you it's not the liver. I'm telling you there's a translation problem, or there's a dual if you look at Homer's Odyssey, this is a different time range completely.
Mm-hmm.
But they hadn't invented the word blue yet.
No, I know. This is why they have also. They
crimson,
said
the sky
it's
is crimson, blood is crimson, everything is crimson. Like, I'm sorry, but there's no human being that walks around in life with a sense of liver.
It's not a sense of liver. It's just where I feel when they would talk about how they feel it. It's like where it is in
butterflies
their body.
In your stomach,
right? But it's not stomach; it's their liver.
It's gut. Okay. Because ours is really stomach.
But they knew anatomy
maybe
enough
it's been
to know
the liver.
The
maybe you have butterflies in your liver when you fall in love. You've always considered it as your stomach, but it never was. If you really think about it, it's always been. So love is in the liver.
Interestingly, they were able to group different emotions according to body regions. Sympathy and Schadenfreude apparently are similarly grouped in the throat and like the testicles. Happiness, pleasure were together, and then you have pride, desire, and love all kind of grouping together. Additionally, sadness, distress, fear, anger, contempt.
All in the pancreas.
Also. Yeah. But the methodology is very interesting. I think the way, of course, take it with a grain of salt. These are ancient texts, and they're trying to compare the ancient text and wording and concepts to what we experience today and how we describe things today. And of course, there's no direct equality, but and today we can have self-reported experience, but this is like, oh, hey, the Mesopotamians wrote stuff down on clay tablets. What did they report? Might be self-reported, or maybe somebody else was reporting it and said that person looks like they have a big liver. I don't know. But the researchers suggest that this could be a useful way to explore intercultural differences in the way that we experience emotions, because of the way that our emotions are not just words. They are actually bodily feelings and we are embodied organisms. And so if we can come to a place where we can compare the way that our bodies feel in particular emotions, that maybe we'll be able to find more similarities or differences. I don't know. But they're going to be looking at a whole bunch of text material from the 20th century with 100 million words, and they're going to examine Finnish data. And they are also looking at ancient Near Eastern empires. There's a whole bunch of stuff they're looking at, and they're hoping that this will be a way to continue to analyze emotional semantics.
Finnish data. How many Finnish?
They feel everything in their elbows. I have no idea. I'm sorry. I don't want to offend anybody.
No, I'm not trying to offend anybody. But my point would be like, yeah. Well, I guess I don't know what my. I guess that would have been my point. Never mind. I should just be quiet.
How does your scalp feel?
How does my scalp feel? I don't feel with my scalp.
Have you ever had an EEG?
I have to look up my tablets to see.
Have you ever had an EEG?
No.
With the cap and the electrodes and the wet gel and all this stuff.
No, I won't let them look in there.
And they're like, oh, I can't get it right. We're not getting good connection, and we got to put more gel here. And then the gel's drying up, and it's taking forever. And they can do one little test, and then if you take the cap off, everything has to be reset, and it's really difficult. And if you have hair, oh my gosh, hair gets in the way of everything. So these researchers just publishing in Cell Biomaterials, these researchers out of the University of Texas at Austin have developed a new method of EEG with temporary tattoos on your scalp. And it's pretty awesome, actually. Although they have only tested it with people who have they have hair, but very short, kind of military buzz cut kind of hair. The idea behind what they are suggesting and what they're developing is they have created a flexible gel that's printable and that can be microjetted onto a surface. In this case, it's your head. 3D mapping of the scalp allows an accurate measurement with, they use 3 dots to calibrate the measurement and how everything is mapped and then placed. And they are able to know exactly what your scalp looks like and set up points where they should put electrodes or points of contact for electrodes for a long-term measurement period. And in this, there's no actual tattooing. It's all a temporary tattoo. It wears off if you sweat too much, soap and water will take it off over 6 hours, and it doesn't seem to work well anymore. But in their models, what they were able to show is that they could 3D model a person's head and then accurately develop EEG application points, which, because of their electrically conductive gel, they could place accurately. And then basically it's just like you get temporary tattoo microjet spray painted on your head, and then they just attach 2 electrodes at the base of your skull, which is where the actual interconnect happens. So you don't have to wear a cap. You can have curly hair. One of the things they say is they still haven't developed a system that can deal with different color skin. So racial bias is still a problem, but that's probably not something that can't be overcome. And they suggest that it needs to be worked on.
Is that for the initial modeling or something? Like, what are they talking about?
For later. So right now, like I said, they've got a person with a buzz cut who had their head measured. Currently, the problem with EEG research is inexact placement of the electrodes because you put a cap on and the cap moves. And if you have hair, your hair holds it away from your head, and there's all sorts of issues. And so what they are
I think it's
suggesting,
quite clear for that. You're gonna have to shave your head for this one. This
no,
is one of those.
no, what they are suggesting is that, like, this is what they're saying. Right now, we have done this thing with someone with hair, but it's very, very short. But
Yeah.
what we are suggesting is that in the future, we could create this technology because of the way that the gel works and the microjetting works, that we could potentially allow you to keep your hair on your head. You don't have to cut all your hair off. You could have curly hair. You could have different kinds of hair, because this microjet technology is like, I'm going to shoot through all of that tangled stuff on your head. It's going to get to your scalp no matter what. Yeah, no.
In the
I mean,
future, it's not getting through this. I'm telling you, I got a
right?
mop up there. Yeah.
But it's the idea is, maybe, and they say that the scalp imaging and the microjet printing doesn't take that long. It's like 15 to 30 minutes. So the whole procedure to set it all up, it takes a little while, but then you don't have to take a cap on and off. There's no researchers going, oh, it's not working, we have to do it over again. It's set up on your scalp. They have the interconnects on the back of your neck, and everything works for up to 6 hours. They had a lady researcher apparently do jumping jacks intermittently for like 4 hours or something to see how sweat impacted all of the material that they've developed, which is biocompatible, and it supposedly doesn't cause any allergies or anything. But I think this is potentially very useful and interesting. I mean, I don't necessarily, if you're going in for epileptic treatments, if you're going in for regular neural stimulation, if you're going in for regular neural measurements of whatever kind, this kind of methodology could lead to a reality where you don't have to shave your head to undergo the treatments that you need to have done. And so you could live a life where you are outwardly very normal, but still having to go to the hospital and have treatments and all that kind of stuff. So I have hope for this. I think it's a very cool technology. I'm very interested in watching it develop, and hopefully it'll go places. And I don't know if it will, but we know EEG works well, but there are limitations. But I do want to share some videos with you that are just very cool. So we've got some videos from the researchers of the printing of the electrodes. These are temporary on the hairy scalp. And when they say hairy scalp, the hair is probably less than an inch. And so they've got robotic machines that are microjetting these dots of tattoo ink, they call it. It vibrates a lot, but they say it is non-contact. You're not putting needles in your head. It causes no discomfort to the patients.
So an interesting application for this then also. I don't know if they're capable of for deep transcranial when they do electroshock therapy type stuff. Is this
right?
Like, yeah, because placement there is also,
it's necessary. It's specific. And maybe there's a combination of things that you could do where maybe you do go in the first time and they scan your head and they figure out locations. They microjet this stuff on and they show that it works. And then you go get a real tattoo and then you let your hair grow over it. And then every time you come back, it's like, easy. There's no problem. Start here.
But then if you did get head tattoos, you may be causing, like, There's one reason not to get yourself a skull tattoo. If you were looking for one, if you were like, I can't decide, this could interfere because there's metal in tattoo ink.
Oh, it depends on the color, but yeah.
Oh, okay. I'm not that. My tattoo knowledge isn't extensive, but my understanding is like that it could interfere with, or it like shows up in MRIs or something. Like it has like a problem in some context.
Yeah, but on the flip side of this, it is if this continues to develop in a way where what they've shown is that they're temporary tattoos, they can be accurately placed because of 3D scanning. They can also, so it's repeatable. You can put your head back in the same harness anytime, and it'll always be the same. And the material is flexible. It doesn't hurt. You're not actually getting a tattoo, whatever. Even beyond that, we could go to further instances of how this could be used beyond the EEG application.
Like a
what other
Parkinson's type?
Yeah, what other body parts could you put these tattoos on for either stimulation or monitoring?
Monitoring and like motor control, like that's one of the things that like there's already things that have been designed for this, but helping mitigate motor control by, I think by electrically stimulating things to a point where they're not being overstimulated from external.
Right, but maybe you don't want to have like a huge wristband on. Maybe if you need something very specific and minimal in terms of its placement and how it's put on your body, maybe, could this kind of system also integrate with clothing that we wear? Could this kind of system, I mean, there's so many things that I see this potentially, like 3D, not just of your skull, but 3D body scans where these temporary tattoos can be placed anywhere on your body. They last for, until you take a shower, and they're not invasive, and they're not these huge things that are clunky and large, and you can find a way to hide the wires and whatever. Or maybe they have interfaces like with the wristband of your sweatshirt or something of that. I mean, there's so many little things that I think are exciting.
In the picture that we've got up here.
Oh, it's got a massive little, like the computer interface in the hanging on his back.
Control with wires and everything hanging off of the person's back. Yeah. And then it ends in these nice little tattoos in the skull. And you're like, really? You could have just continued the wire for a couple more inches and you wouldn't have needed the tattoo thingy. But
no, but this allows for long term in the sense of several hours of monitoring. So sleep studies. I think that's one of the interesting ones.
That's brilliant because that would be, if you were wearing like the skull cap and the thing, and now it's interfering with your, your rest. That would be very.
Or you did a sleep thing and you ripped it off your head. Suddenly, all the data's gone. Yeah. So something like sleep studies, this could come in very handy. And if they figured out how to allow you to not shave your head, that'd be cool. So everyone, as you go to sleep, think about the fact that you don't have to have little tattoos, temporary tattoos printed on your head by very accurate microjetting tattoo robots. Have we done it?
Have we come to the end of yet another show? Is it possible?
Yeah. Did you talk about all your stuff?
There's other stuff, but nothing that we won't get sued over.
No. Oh my goodness. Well, everybody, I thank you so much for joining us for another episode of This Week in Science. I hope that you enjoyed the show. Yes, Paul Disney, we scienced successfully. Thank you so much. We made it through. We made it through the science-ness. Somehow we made it through. I put a temporary tattoo on my head, did you? All right, everyone. I would love to give shout outs to Fada. Thank you so much for your help with show notes and social media. Can't get anything done without you. Gord, thank you. Arnlore, thank you. Everyone in the chat rooms, thank you for keeping the chat room cool. Thank you for thinking and for commenting and bringing your ideas and questions to the room. Rachel, thank you for editing the show and Identity 4. Thank you so much for your work to record the show. You're there all the time. You've been helping us for so long. And all of you who have been helping us for so long. I appreciate you sticking around and hanging out with us. So, Patreons, I want to thank you. Thank you to Eden Mundell, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Richard Badge, Bob Coles, Kent Northcote, George Corus, PL, Vera Zarb, John Radiswamy, Chris Wozniak, Vegard Shefstad, Donathan Styles, a.k.a. Don Stylo, Ali Coffin, Reagan Shuburu, Sarah Forfar, Don Mundus, PIG, Stephen Albaran, Daryl Myshak, Andrew Swanson, Fredis 104, Sky Luke, Promorovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Seema, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, John Burton, no, not John, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artie Mom, Greg Argyum, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkan, Kevin Perishan, Aaron Lutham, Bob Calder, Marjorie Paul Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And I do want to give a special shout out to Matthew Calabro, who wrote in and to say happy Thanksgiving to all of us and said, I appreciate all that you do for the community and more specifically for me this year. You helped brighten the path along the way. Thank you. Thank you, Matthew, for listening to the show, being a part of TWIS and everything that we do here. We appreciate you. And we appreciate everyone. So thank you. Thank you so much. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, and again, Thursday, 5 a.m. Central European time, broadcasting live from our Twitch, YouTube, and Facebook channels.
If you want to listen to us as a podcast, just search for This Week in Science wherever podcasts are found. You'll probably find us. If you enjoyed the show, please get your friends to subscribe too. They need us. They really, really need us a lot more than they need Joe Rogan or Andrew Huberman.
Oh, gosh. Oh, do people need that?
No, they don't need that. They need us. That's what I'm saying. We need some groundswell, everybody.
For more information on anything you've heard here today, show notes, links to stories will be available on a website. I don't know which one. Oh, I do. It says it here, www.twis.org. You can look up all of the links to the stories and then find the actual research paper and then read up on the research paper. And then you can Google that subject and something like Google Scholar and find more. You can learn about rabbit holes. Those are also there on the website.
And you can sign up for our newsletter that we might send someday.
A newsletter.
We love your feedback, so send us a note. Send us a thanks. Send us a what. If you want us to cover a topic or think about some things some more, let us know on one of our social media accounts. TWIS at TWIS Science is usually what we are, or send us an email. I mean, that's really a good way. But make sure you put TWIS in the subject line so your email does not get spam filtered into the calcified remains of a Neanderthal handprint. It'll take too long.
We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.